Okay, Bill, that was the introduction in Dutch, just the rules of the game that we that we decided to do.
I didn't
Um,
understand a word other than I did hear my name. That's all.
okay, well, that's sufficient. Um, well, it's a great honor for us, and I think for all the participants tonight, members from the Dutch Society of Psychical Research, and other invitees interested to be an audience for you. And we're looking really forward to your presentation. Um, just let me give a brief introduction. I did give an introduction in our invitation. Um, Professor Bengston is emeritus professor at the St. Joseph University in New York, which is resigning now. He was president of the Society for Scientific Exploration, which also gives a journal, of which he also was was an editor. Has done research over many decades for anomalous healing, particularly about curing cancer in mice. We will certainly talk about that, but it's not limited to only to these small animals, but it goes beyond that. Um, but I think Bill will tell us much more about the methods, the research, and the results and possible consequences. So I hope that. So I propose to leave the floor to you now, Bill.
Thank you, and I appreciate the invitation. I didn't hear the entire introduction in Dutch. Actually, I heard it. I didn't understand it. The entire introduction in Dutch about the format for this, and Paul and I have been talking about letting me prattle on for a while. But since it's a nice small group, if I say something that you find completely outrageous or you can't go with or something like that, and you got to ask the question. It's okay. It's really okay.
Yeah,
Uh,
there's also a raise hand possibility
yeah,
in the Zoom, but you might also attempt to raise your hands in front of the screen. But if that doesn't work, there's somewhere a button called raise hand.
yeah. And the other thing is apparently there's a chat room someplace or other.
Yeah.
I won't be checking the chat room, but maybe Paul or somebody can say, oh, there's an interesting question. Let's pass it on and do it. So I don't intend this to be a seriously formal presentation. I hope it to be seriously interesting, but I don't know how formal it's going to be. So I'm going to share my screen. Let's see what happens here. And if this works, I'm always amazed. Are we good? That's good?
Yes.
Okay.
hmm
Who knew it would work? Okay, so the basic idea, obviously, about today or tonight where you are, is how do we make healing conventional? And so I've been studying healing for, I don't know, 70, 80 years, something like that. And it's interesting. We know quite a bit about it. We know that we don't know very much. That's an interesting thing to know. And we've done a whole lot of experiments, et cetera, et cetera. But it's still fringe. It's still an anomaly. It's still when healing happens, you go, oh, "What is that about?" And you're startled. And so it's always there's this cloud, if you will, of it's not conventional. Give me something that's off the press and legitimate and everything else. And so my question now is, and the focus for tonight is, can we turn healing into something that is conventional? And I'll give you a general idea, if I can find the. This is the bottom line. So if you want to leave, leave. If you're going to make healing conventional, it needs to be storable and it needs to be scalable. If you have storability without scalability, you got nothing. If you got scalability without storability, you got nothing. And so I'm going to focus on the two parts. Can healing be stored and can healing be scaled? And to give you the bottom line, yeah, the answer is going to be yes. It's not going to be quite that simple, but the answer is going to be yes. So if you think of healing and the way a lot of people think of healing, you think of a healer and a healee. And why they have their eyes closed, I have no idea. But This is something just pulled off the web. We have a healer and we have a healy, and this is fine. And let's assume that we study this until our brains fall out. And we find out that, yes, in fact, something is happening in healing. And let's say we find out that, this is the physiological change, and we find out, and we find out, and we find out. That's all beautiful. And, I've been doing this thing for over 100 years. I understand the impetus behind the questions. But this is very different from saying, can we make this conventional? And conventional would mean I got to go out and I need a healing. I got to go out and I need the way that you would in any other field of medicine. How can I make it so I can go out and get a healing? And not just find somebody who's willing to put their hands around them or on or near or something like that. What are the parameters and what are the ways we could possibly do this? That's what we're up to tonight. So we're used to the idea of storability, clearly. It's all over the place. We just don't think of it in terms of storability and scalability. So if we take electricity and we put it in a box, what are the odds? Think back, probably most of you are under 300 years old, but about 300 years ago, if you had come along and said, I think I can capture what's happening in lightning and put it into a bottle, they'd think you're out of your mind. But electricity, charge, etc., has been captured and it has been stored. And in fact, it's been done so many times, we don't even think about it. And if it can be stored, then maybe it can be scaled. So it's interesting to study a battery, but can we make a boatload of batteries? That would be the scalability. And we do this in physics, we do this in chemistry, we do this in biology, we do this in medicine. This is probably fish oil. So you got fish oil and you can go out and eat a fish, or you can go out and you can buy a fish oil that's been stored, and apparently fish oil can be scaled. So there's distribution systems all over the place. You can go out and get yourself some fish oil if that's what you need. Can we do the same thing with healing? So this is the plot. Again, I like to give away the answer as we start. I'm going to talk a little bit about some previous healing studies very quickly. And then we're going to look at some studies on can you store? And the answer is going to be yes. And so we've done controlled systematic studies that healing can be stored in organic material. It can be stored in inorganic material. Healing can also be recorded. And I'll show you some experiments on that. And the question will be, if we record it and we play it back, do we reproduce the effect? If I store it in a battery and I reproduce, yes, the battery works. We can scale the fish oil. Can we scale the healing? So we're going to look at some organic studies on organic material, studies on inorganic material, and we'll talk about the scaling and storing problem, and we'll take it from there. If I can find out where I am. That's not where I am. Okay. So since I've been doing this 132 years, this is the general area, the goals, the kind of places I like to play with. What are the parameters of healing? What makes it work better? What makes it work worse? What can it work on? What can't it work on? So you can take obvious parameters. Distance. That's a physical parameter. Does distance matter in healing? The answer is going to be no. So if we're going to understand healing, we have to understand something that distance doesn't apply to. That's an interesting question. Now, if distance doesn't apply, it doesn't diminish with distance. About 4 seconds reflection would you'd come to the conclusion, therefore it's not energy. There's no energy in energy healing. That doesn't take a lot of deep thinking. Yet a lot of people on the outside talk about energy healing. I actually have a book called The Energy Cure. Wasn't my idea for a title. Publisher said you got to do that. So what are the physical parameters of healing? What are the psychological parameters of healing? Does it matter I'm just making stuff up. Does it matter, for example, whether the healer believes? Does it matter whether the healee believes? Does it matter whether, does it, what matters? Should I focus? Should I put my attention on, put my attention off? Those are parameters of healing. Oh, just a quick blip at that. All other things being equal, see, we know something about this. All other things being equal, which is a statistician's cop-out. Fortunately, I'm a statistician, but all other things being equal, does belief matter? And the answer is probably a little bit. A little bit. Believers don't heal as well as disbelievers, which is pretty interesting. So belief, I think, retards healing. Not so much, but a little bit. It's not nothing. Oh, I'm interested in the physiological correlates of healing. What happens to the brain? What happens to the spleen? What happens to the body? What happens? The scientific mother lode is the development of a theoretical model. I don't have one. Meaning, I've been doing this 140 years. I don't know anybody who knows how healing works. I got a lot of people who say they know how healing works, and they can even speak definitively. But about 4 seconds of counter hypotheses to some of their ideas, and their system crumbles. I don't know anybody, including, or maybe especially me, that understands how this stuff works. But in the interim, even if we don't understand how it works, can we make practical stuff out of it? So can we move, for example, from a science model where I'd like to understand how the plot works, and can I move to an engineering model, and I'd like to see how do I make stuff work better? And we don't have to understand something to make it work. So if I put 10 theoretical physicists in a room, which I can do, there's a boatload of them at the SSE, and I ask them a simple question, what's gravity? Well, someone's going to think there's graviton, some are going to think there's curved space, some are going to think there's this, some are going to think there's that, some the other thing. And at the end, they get into a fist fight. Now, nobody disagrees that there's such a thing as gravity. They just disagree about what it might be. Everybody should be very skeptical if somebody has an idea about how this stuff works, but it doesn't mean it doesn't work. And the question that I'm dealing with now is, can we turn it into a technology even if we don't understand it? See if I can go to the next one. I don't know if I did that right. I'm lost here. I'll be with you soon. Okay. So I'm going to show you really brief. This is the general plot. I work in labs and clinical studies and all that stuff, and I also I've done a lot of mice work. It's not everything I've done, but I've done a lot of mice work. I just finished my 19th and 20th mice experiments at Tokyo University, for example. So I've done a lot of experiments, and here's a general idea. These are mice experiments, and the mice experiments on a mammary cancer, I've done 2 experiments at City University of New York. I've done 2 experiments at St. Joseph's College. I've done one, and so you get the idea. And lately I've been working out of Brown Medical School. So I've done a lot of experiments on cancer. That's not all I do. I've done a lot of experiments on mammary cancer. I've done a lot of experiments on sarcomas. I've done studies on oncogenic mice. I've done studies on nude mice. I've done studies on extremely aggressive, and I've done comparative studies, breast cancer, bladder cancer, melanoma, and in terms of healing response. I got a lot, I mean, there's a lot more, but you get the plot. So I'm not pulling this stuff out of the air. I'm doing really systematic studies. I've got 2 articles out for peer review and publication. One was just accepted. Second one I'm waiting to hear on. And so the experiments are going on. This is an active research agenda. And what I do is I think in very, very simple terms. If I'm going to try to unravel something as complex as healing, I want to get into an area where people already think they understand how the world works, and then I add a single variable, which will be healing. So, for example, if we take a mammary cancer in a mouse and we get it out of Jackson Labs or the National Cancer Institute or we know exactly what we're studying. We know the tumor type, we know the strain, we know all sorts of stuff. And this particular form of cancer, this experiments on mammary cancer that I'm going to show you just a couple of ugly pictures. This is probably the most studied cancer model in animals ever. There are literally a couple of thousand publications on this one mouse alone. So it's been studied. We mapped it out. We know what's going on. We know each step. We know the stages. We know if we inject cancer into this, we know exactly what's going to happen, and we know what's going to happen next. That's a beautiful thing. And then I come along and I interject a single variable, healing. What if we take this mammary cancer model, thousands of studies, and we add healing to it? What will happen to the outcome? So nice and simple. And if we already know the stuff, the difference is whatever we've interjected in the healing studies. So that's the general plot. So here's an example of a mouse that's not in good shape. This has been injected with the mammary cancer. In the mammary cancer, 100% of the mice die in a month. It's ugly research, and 100% of them die. 100% of them die. That's got to sink in. In thousands of publications, 100% of the mice die. And the distribution looks like a normal curve. Most die within this day and that day. Fewer still on the outskirts. But we know what's going to happen. And so it's an ugly way to go. It's a non-metastatic cancer, and it just kind of grows and it saps the life out of the mouse, and they'll die within a month. And so this is known, this is studied, this is repeatable, this is anything you want. So we come along and we introduce a single variable. And this is a picture of me at Brown University. This is before COVID, incidentally. I wasn't dressed for COVID. This is because you need the labs to be clean and sterile and all those kinds of things. So in there, introducing a single variable. So we got a bunch of mice. We know what's going to happen with the different strains of mice, and we add healing to it, and then we see what the difference is. If you don't do anything, short version, all the mice die. All the mice die. If you do healing, something interesting happens. The tumor grows, and then it starts to ulcerate. And the ulceration can get pretty severe. So here's a picture, and here's the ulceration. But look at the coat, and you can't see the eyes, but look at the coat. This mouse is otherwise healthy. It's going through a process that we've never seen and has not been seen before. It's going through stages that nobody's ever seen before. And the end result of these stages is a cured mouse. Now, not a remitted mouse, a cured mouse. This will be a mouse that's cured, and it's cured for its entire lifespan. And it's even farther than that. If we re-inject it later in its life with cancer, it can't get cancer again. Now, that's reasonably interesting. But it goes through phases and stages that I never would have dreamed up. I mean, that looks ugly to me. But it turns out that's a necessary process in the cure. And we know what will happen after this. And we know what will happen after that. And we know what will happen because we've done a boatload of studies before and then after healing. So we know quite a bit from quite a bit of studies. And I can't talk about these because this will go on for days, but we have reliable empirical data. And so I'm pretty conservative in terms of what I'll say. And if I'm going to say something, that means I've reproduced it at least twice in independent labs. So I'm not just making stuff up and going, ooh, you want to see a weird thing that happened. This is stuff that is extremely systematic. So if we're talking about the cancer experiments, we can say, without quantifying. Cures abound. They're all over the frickin' place. So we have hundreds and hundreds and hundreds of cured mice. I can say with some confidence, or it wouldn't be up here, that distance doesn't matter. So if I do a study and the mice are 2 inches away, and then I do another study and the mice are 2,000 miles away, or the mice are 5,000 kilometers away, it doesn't make any difference. The effect is the same. Now, what in heaven's name doesn't diminish with distance? And it's certainly not energy, because energy diminishes with distance. So you tell people distance is irrelevant, and they go, yeah, of course. That's a dime a dozen. Yeah, and that's true. And we have it, and we test it, and we can do it, and we can reproduce it in a lab, and. But that means it's not energy. So what are we looking for? That's the question. I can tell you there's a dose response. It's not simple, but there is a dose response out there. I can tell you it's not a field effect. We're not like changing the world. If I put mice in 5,000 kilometers away, I don't cure all the mice between here and 5,000 kilometers away. It doesn't go out as a field yet. It hits a target. What in heaven's name would hit a target and not go out as a field? Belief is irrelevant. And I should keep trucking here. Placebo effects in mice and cell cultures can be demonstrated. I have experiments that show placebo effects in mice that exactly mirror that of people. I have experiments that show that placebo effects in cell cultures. I even have a study of placebo effects in placebos, but I'm not going to go down that road right now. Placebos are not psychological. There may be a psychological component, but they're an actual physical resonant bonding of spatially separate units. Healing is not a PK effect. I had to say that to a parapsychological group. It's not mind over matter. I thought it was at one point, but until I started my experiments, I went, wow, this isn't PK. And it's certainly not happening the way I expect it to happen. It's certainly not happening the way I want it to happen. We're discovering replicable patterns out there which occur when you interject healing. It's not PK, and it's not your wishes that transform the universe and so that it comes out according to your wishes. I can talk about healer and healees exhibiting brain resonance and but these are separate talks. The more interesting task is to list the stuff you don't know. And that list would go on for years. So is healing energy? Is it information? How does it work, for crying out loud? How do distant treatments hit a target? How wide do different techniques different outcome? How come a healer A and healer B using the same technique have a different outcome? How come technique A gives a different result than technique B? I mean, nobody has any idea. Can we measure this stuff? Can we make a charge-o-meter? Can we make a heal-o-meter? How about multiple healers? Is there a linear effect with multiple healers and the effect size? Does healing come primarily from the healer or the healee? I can keep going. Bottom line is, I don't know very much in comparison to the things I don't know. And I don't even have a good list of the things that I don't know that I should have on the list. But I can tell you that when you test your ideas, you'll find them almost always, this is a beautiful thing, to be wrong. You don't know anybody who's been more wrong than me. Because I keep testing and I go, well, that's new. Well, that's different. That doesn't make sense. I must have no idea what I'm talking about. No, I really don't. I really don't. So let's get to the heart of the matter. Can we store and can we scale? That's the question. So I'm going to do a couple of quick, without technical detail. I can go into it if you want it later. We've done a bunch of stuff on cotton. So you take an ordinary piece of cotton. I don't have one with me. I have a napkin. Let's pretend it's a piece of cotton. So if we have a piece of cotton, can healing itself, what if we treat the cotton? Will the cotton, in fact, reproduce the healing as if the healer was doing the normal thing? And the short answer is yes. And this doesn't come from me. I didn't discover this. This goes back, I think, in terms of systematic study, to the great Bernard Grad at McGill University. And he's the guy who is the granddaddy of all, I think, healing research. You want to know the granddaddy, go read some Bernard Grad. And Grad demonstrated that cotton is a good holder of the information involved in healing, and it actually seems to, using this metaphorically, store it. Now notice, if you have a piece of cotton and it's stored, it doesn't necessarily mean it's going to be scaled. It just means it can be used as a storage. Can you scale it? Separate issue. So we're always going to be asking, can this store? Can this scale? So without going geeky, we do genomic studies, and we do this and we do that, and we do the other thing. And we find out what happens if cotton that's been treated with healing intention, if cotton is moved near cancer cells, as they get closer to the cancer cells start to genomically flip. If the cotton has not been charged, they don't flip. So in other words, a cancer cell, using this loosely, can recognize whether cotton has been treated, indicating a mechanism of indicating storage. Other organic materials, cell medium. We do experiments. Some of these were done at UC San Diego. You take the cell medium, which is the nutrient bath that cells live in if you're doing cell studies, in vitro studies. And we take we don't treat the cells, I mean, we do that too, but we don't treat the cells, we treat the cell medium and then see if there's a change in what happens to the cells grown in the cell medium. We already know the answer. The cell medium stuff is used all over the world. I'm sure it's in the Netherlands. It's exact what I have up on the screen, because it basically, this company supplies the world with cells stuff. So what happens? We already know that. But what happens when you interject healing? You get wild effects. And by wild effects, I mean as much as a difference in growth rate of 600%. That's not subtle. That's in your face with a 2x4. Now we go back to, well, let's keep trucking.
Just one interruption, Bill. To be sure, these media, you treat them exactly as you would treat the mice or other Heelys?
Yes. So essentially put the hands around, and then we do the cycling stuff that we do. And so we cycle on this stuff, or we don't cycle on the stuff. So we've got controls, we've got the ones that have been, we've randomized, all that stuff has gone on. Wild changes, wild effects on the cells. Now, I'm pretty sure that the cell cultures don't believe. It's not a psychological effect. There's some real physical effect going on here, and it's not subtle. And we take, these are supposed to be mice. These are pictures pulled off the web. It's actually a picture of a rat, but it's supposed to be, it'll tell the story. Mm-hmm. So we've got over on the left, a treated, ulcerated mouse, like you saw before, except you were looking at a mouse. You had an ulceration in its side. What happens, what's going on in that ulceration, and does it contain the information of the healing, is the question. So what we do is we take some blood out of a treated mouse, and we inject it into an untreated mouse, meaning no human has tried any healing. And what happens? Everybody's cured. Now that's reasonably interesting. You can see the analog to a vaccine. There's something in the mouse that's been treated, very likely related at least in part to the immune system, and it's transferable from one mouse to another. Think vaccines. You can store it, you can scale it. Could we make a healing vaccine? My guess is yes. I don't know that anybody would allow us to use it, but my guess is you could make it. I think we know enough now to make it. Now, that's not cheap to do, and that's a very separate issue from will anybody allow us to use it? So these are just possibilities. Now we can move to inorganic stuff. And by organic and inorganic, I don't mean does it have pesticides. I mean, does it have a carbon atom? So there's no carbon atom in water. So it's inorganic. And we take water bottles and we treat the water bottles themselves and they're fed to mice we never meet. And they all get cured. So again, we have now storage in a variety of organic materials, at least one type of inorganic material. We have storage in life itself. I mean, you get the idea, healing can be stored. The question is, what's the range of media that it can be stored in? So we still have the dual question, can it be stored? Can it be scaled? Again, it can be stored without being scalable. It can be scalable without storing the information that was in the original. In the perfect world, storable, scalable. So we went with, or my agenda started, let's go with the most scalable and see if we can store it. And what's the most scalable way that you could store healing intention? I mean, you can't get more than this, recording it. So can you imagine if we could record healing intention? If you can record something, given the infrastructure of the web, you can make it global. Now that's scalable. So if I had a recording that reproduced the original and I scaled it, I'm scaling it globally. If I have cotton and cotton stores healing information, it does, that doesn't mean it's scalable. If there's a way to scale water, then it might be scalable. If I've got water, same thing. If I've got water, and I use, as I've demonstrated here, that you can store healing intention in water, it doesn't mean it's scalable. Can we come up with something that demonstrates whether healing can be scaled and stored in water? So I started out a few years ago saying, well, we can't get more scalable than storage. So if we can storage and kill that problem, we've got it licked. So you can be any place in the world, say, I need a fix for, and you get the fix. That's scalable. So my recording team, the guy second from the left, you probably recognized coming from parapsychology, Dean Radin. So I got on the left, John McMichael, Dean Radin, Mike Turner. He has a PhD in signal analysis. So I brought him in on the thing. And Sarah Besum does most of the lab work inside. So the general idea was this. Let's make sure we got a clean environment. So we got a nice, clean Faraday cage. It's also big. So this is a big walk-in Faraday cage. And the Faraday cage screens out electromagnetic interference. So we're going to have a pure signal. And it's a big, heavy thing. And inside, which is all dressed up and such, and we put a bunch of detectors. We've got magnetometers, and we even have caduceus antennas, seeing if we can pick up scalar fields. So we got a bunch of stuff, and we put a bunch of stuff into the Faraday cage because we don't know what to do. We don't know how to record. So you start by throwing the kitchen sink at it, and we got actually 33 different detectors in the Faraday cage, all running as inside people are generating, so to speak, healing intention. Can we record it in this safe room, is the question. So we got 38 signals, or 38 detectors in there. We're collecting a lot of stuff. So we make a recording, and we put 3 people inside the Faraday cage. And what they actually did was they held pieces of cotton and did the healing technique that I use. And so we had 3 people inside the Faraday cage doing this, and 38 detectors are going on. What are we detecting? I don't know. But what happens if we do that? That was the experiment. So what happens if we then take this recording and we play the recording back to some cancer cells, what happens? So here we have an amplifier. This is just taken from Amazon. We got a couple of speakers that we bought from Amazon, and we bought them because they fit in the incubator. And we have a bunch of cancer cells which come out of another lab. But so we ordinary audio stuff being played to cancer cells. Cancer cells, as far as I know, don't believe anything. They can't hear the recording. Actually, it's not audible to the human ear. And so we play through ordinary off-the-shelf speakers and amplifier to cancer cells which have been prepared in vitro. And we play it for different lengths, 4 hours, 24 hours, 48 hours. We try this, we try that. And we play it to a boatload of cancer cells, and we do a boatload of experiments and a boatload of genomic studies. And the short version is we looked at 167 genes, and 68 of those had significant genomic changes which were reliable. And we did this 7 times at Brown University. So this is what I mean by it's a reliable effect. It's not something, ooh, do you believe, or something like that. And so we've got 68 genomic changes, which have to do with upregulation of apoptosis, which have to do with telomere length, which have to do with immune response. And it becomes reasonably interesting. In fact, it was so interesting, we published this in a conventional biology journal. Go figure. And in this playing of the recording, which is maximally scalable, to cancer cells, we came to some conclusions. First of all, something happens, and something happens repeatedly, and something is happening. I mean, it's mathematically so unlikely that something happens. And if something happens, we've captured and we've released, think battery, we've captured and released, which means a technology of healing is possible. Now, if a technology of healing is possible, what's the limit? And the answer is, I don't know. But a technology of healing is possible. Now that's interesting in and of itself. And if it's a technology of healing, we can put it out there, make it public, and people can work on it, as you can with any technology. So maybe we've got the first car off the first assembly line, and there's some room for improvement. Well, yes, and we get to today. Yeah, cars are a little bit different than they used to be, but the plot is the same. We're just improving the technology. So can we have a technology? Yep. Now here comes the kicker for us. We did comparative analyses of how long genomic changes take and how many protein folds and this and that and the other thing. And it turns out the recording of people charging cotton inside of a Faraday cage, that recording is not as strong as doing healing live. Something's going on, but we haven't captured the whole thing. So in other words, we can't pack up and go home. Just make it worldwide applicable. You're going to get something happens, something very significant happens, but it doesn't match doing it live. So if you put your hands around a bunch of cell cultures, you're going to get a different effect than if you put a recording up to the cell cultures. Something happens in both, but I would still go with the hands-on or, it's analog before I would just give it, I would say we have not succeeded. We've succeeded in the statement of principle, but we haven't succeeded in capturing it. We don't know why that is. Is it because it's a digital signal and healing is analog? Is it because of the sampling rate of the digitization? Is it because, we don't know. Or we just had the wrong detectors, or we're asking the wrong question. We just may have no idea what kind of question we're really asking. And this last one didn't make it into the paper because it's too out there. We didn't think we could get published if we put it in there. Apparently, healing activity produces conditioned space. So the phenomenon occurs in the physical space even after the stimulus has been turned off. This is also reasonably interesting. It also has one or two implications, not only for experimental design, but also for clinical practice. The way this was discovered, which is the way most things are discovered, which is by accident, the way this was discovered was a bunch of super sensitive people, you all met them, listening to the recording and trying to get a sense about what's going on in the recording and giving me feedback because they're the super sensitive type. And then I would turn it off and to a person, they'd say, it's not off. And I'd say, what do you mean it's not off? It's off. No, I can still sense it. It's here. You know, I'd even pull out a plug, hold it up and go, it's not even plugged in. They go, no, it's still going. So I called up to Providence, said, here's an experiment for you. Play the recording to an empty incubator, wait a long while, and then stick some cancer cells in there, no more recording, and see if anything happens. And dang it, they were right. It's still playing. Even 3 days later, you can stick cancer cells in the incubator, no recording going on anymore, they'll genomically flip. Now think of the implications of that for doing controlled experiments. They're pretty profound. Think of the implications of that for clinical practice. They're pretty profound. We just don't have a chance to do it, not now. So we take the recording and what can we do with mice? Let's try the short version. Let's try, play the recording. These are the same speakers, the same amplifier, the same everything, same computer running the stuff, except now instead of cancer cells, we have cancerous mice. What happens if you play to cancerous mice? And we ran into some problems with the animal ethics people at Brown, to my consternation, and lost a lot of money. But the short version is this. We're starting to get an analogous effect in cancerous mice that had never been seen before. And the tumors were growing, ulcerating, and we don't know whether we would have taken it to full cure. But we ran into some problems with the animal ethics folks who came in and canceled our study about a little more than halfway through. Even though the mice seemed healthy, coats were clear, eyes are clear, eating, drinking, having a good time. The animal ethics people shut us down. So we never finished the analyses, though we did have a pretty good paper that also went to a biology journal. And we could say with confidence that playing the recording to cancerous mice, there are biological effects. And some of them are task specific, such as an increase in monocytes and this, that, and the other thing. And some of them are also not subtle. Look at the last one, if I can find the pointer. I can't. Spleen weight increased 5-fold. So this is an enormous, their spleens kind of went up when they heard the recording. Now, what does hearing a recording have to do with spleen weight going up? I mean, in your healing, you're not trying to increase spleen weight. And you get the idea. So there are natural processes where we're trying to discover the patterns of healing that are out there. So after quite a bit of time, was able to find another lab in Tokyo that would, we think, allow us to go all the way through or farther through the experiment. And we tried then with a new twist, comparing bladder cancer, breast cancer, melanoma cell, do they differentially respond to the recording? And the short version, this paper is out for review right now, also in a traditional biology journal. But same idea with the speakers, same idea with the setup. And what we found is, essentially a replication of the effects on the breast cancer model that we had found at Brown. But we didn't, in the melanoma, we didn't see the effect. And so there were no significant effects in the melanoma. Now, the obvious question is, why is that? So tumor volume and mass change in the breast cancer, but they don't change in the melanoma. And we had some weird stuff happen with the bladder cancer model in Tokyo. And that was, they didn't develop tumors. And we did it twice, and they didn't develop any tumors. Now, we don't know whether that means because our healing was extraordinary, or it means that something was the matter with the cell line, even though we had pre-tested it, pre-run, preliminary tests on all the stuff. Um, we don't know what's going on, but this is now out for review. We'll see what the biology journal does to this. So let's go back to a new substance, water. Actually, it's an old substance, but now we want to ask the question, can it be scaled? So we know that water stores. Can we figure out a way to scale it? Can we enhance the treated water? And so we've played with a lot of different methods of doing this. And it turns out, this is the very short version, that if for those of you who are familiar with homeopathy, that you have increased dilutions. So the less stuff you have left in the water, with increased dilutions, you have increase in potency. And I'm not a homeopath. Homeopaths also, some homeopaths in some schools of homeopathy I found, they think that they can scale so you can go and get a homeopathic remedy that's been mass produced. Well, can we take something that's been stored and mass produce it? You can see the overlap. So could we take our treated water, treat it to homeopathic dilutions and succussions, and see if anything happens or changes in the water? The difference between us and the homeopaths is that the homeopaths are starting with a real molecule, whereas we're starting our water studies with water, and then we'll dilute and succuss water itself. Now, I hope that if you really understood what I just said, your head would explode. But, interestingly enough, it works. So we played with many, many, many things in an engineering way because we were not really sure what we're doing. And we settled on a promising bunch of dilutions and succussions to originally treated water. So we start out with water that's been treated as the exactly same way that we've done in our experiments. Then we've taken the water, diluted it, succussed it, and we made a mechanical technology which we were testing to see if it will scale that diluted and succussed water. So once again, the steps. Treat water as before, because we know that it can be stored. And then put it through dilution and succussion, serial dilution and succussion, and then put it through a gizmo that will mass produce that and reproduce it. And we settled on a promising bunch or combination of dilution and succussions, and we call that stuff restore. So this is not a sales technique. I'm just saying the stuff that, let's say you wanted to get some in the Netherlands, you can get it, but it would be called restore. We found a different dilution, succussion, and preparation method for something we call perform. And this is for short-term performance. Restore is a general well-being kind of a thing. Perform is for endurance and strength, things along those lines. And recover is an antiviral. So the short version, we have three, and this is not sales. Trust me, I'm not selling. We have something called perform. We have something called restore. We have something called recover. If you want general health, whoops, going nuts. Yeah, I'm going the wrong way. We'll get there. Okay, so if you want something for general health, you want something for virus problems, or you got something you just want to be short-term better athletically. And we started a course, because I don't believe any of this stuff, we started a course doing systematic studies, now clinical. So we've got the lab work done in water, and we've got the lab work done in this, that, and the other thing. But we started out with the restore, the generic one, treated water that's been diluted and succussed. And we did 3 pilot clinical studies. They weren't experiments, they weren't sufficiently controlled, but they were pilot clinical studies with about 100 volunteers each. We did 2 in the States and 1 in Europe. And we got people with a variety of conditions, a whole variety of conditions, and they volunteered to take 2 of these drops under their tongue 4 times a day for 8 weeks. And we monitored them at baseline, 2 weeks, 4 weeks, 6 weeks, 8 weeks, and they were self-reporting. So we don't have, concurrent medical exams. We simply have, on a scale of 1 to 10, here's how I felt today. 2 weeks later, on a scale of 1 to 10, here's how I feel today. And so we were tracking changes, and did it depend on the kind of condition you had? Now, the results of all 3, so this is about 300 people, were startling. They're not were startling, they are startling. 98% of the people taking Restore for 8 weeks reported improvements in their health. Now, that's a crazy figure, and skeptic in me says, how can this be? It's too much. And we found that we had physical indicators or dimensions, we had mental indicators and dimensions, we had spiritual well-being indicators and dimensions, and we found increases all across the board. And so the conditions which reported positive changes, including cancer, arthritis, depression, anxiety, pain, Parkinson's, you go on and on and on. And it didn't seem to just be a physical thing. We had people who were seriously depressed. We had people on bipolar medications get off it. We had. Now, among the interesting things is this is going to be the inverse of the water. I'm sure of the recording. And the recording, maximally scalable, but we didn't capture something. In the case of the water, it's less scalable, but it's still scalable. And the interesting thing we're looking at now, and I got to take it back into the lab, is the water seems to actually have been enhanced. So in other words, the effect of treated water seems to be stronger than the original treated water. And this, I don't know what to do with this either. So for example, take the last bullet point here. Some effects like Parkinson's appear to be stronger than the original. We've treated a number of Parkinson's patients using, ordinary healing and haven't had great success. It turns out we're better at taking things away from people who have stuff there that they don't want than we are at stimulating things that are missing. In Parkinson's, the brain is not putting out something. So can we stimulate it to put out something? And so far in the healing, not so much. I mean, we get symptomatic improvements in this, that, but it's nothing to flop on the floor about and get all excited. It's not, we don't have a mother lode kind of a thing going on here. In the case of treated water that's gone through this dilution and succussion thing and given to Parkinson's patients, differentially, we don't have enough bodies, to speak definitively, but there seems to be an enhanced effect. And we've had, this is just an anecdote of one. We've had, for example, an astrophysicist who'd been in a wheelchair for 5 years, take the stuff under his tongue. He's gotten rid of the wheelchair and he's walking a mile at a time. Now that's crazy. I mean, I understand that's crazy. The studies go on. Perform, for the athletic stuff, we randomly, blindly give this to athletes. They measure their oxygen intake, if they're weightlifters, how many pounds they can lift. They don't know what they're taking, so they're getting with control groups and all this kind of stuff. And something seriously is going on with Perform. I mean, I understand I'm starting to run out of time here. We've done experiments on plants. We've done experiments on tulips, for example. We're about to do studies on blueberries. We've got a whole lot of stuff going on with plants. If you take the Recover, if you take the Restore, very interesting things happen to the plants. And we've done this on large scale in greenhouses. We're about to go to South Africa to do some stuff. I just finished and just got accepted for publication a study on will these drops have an effect on COVID? And this was a serious double-blind, placebo-controlled, randomized study that I don't think has any methodological flaws or even shortcuts that were made. And the very short version of this is COVID patients entering a hospital for treatment, so they're not casually sick. If they're going into a hospital, they've got COVID. And we've got PCR tests, and we've got. So we've got 160 volunteers, half of whom were random, and this really was a randomization, were randomized to be treated, and half of them were randomized to be controlled. Everybody going into the hospital were volunteering to be part of this. You're going to get sublingual drops. Your physician's going to give it to you. Your nurse is going to give it to you. And you're going to be taking these drops 6 times a day for a week. That's it. And we don't know. You're not going to know. I'm not going to know. The physician's not going to know what's in the bottles. And some of the bottles had ordinary water. And some of the bottles had this special water. And everybody took the drops. Nobody knew what was in them. And then we compared on a variety of measurements symptomatically. Is there a difference between treated and controls? And we found, this is seriously significant stuff. General feeling, how are you feeling? Huge difference in how you're feeling if you took the drops that have gone through our hocus pocus. If you just take drops of water, you're in a hospital being treated. The control group here is not no treatment. The control group here is people getting medical care. So you got a gaggle of people getting medical care, some of whom also take special drops, and some of whom don't take the special drops, they're just taking drops. And nobody knows what's going on. What happens to the general feeling? How do you feel? And we follow them daily across time. Huge difference, very to many decimal places, significance. Their temperature goes down. Their sore throats go away. Their coughs. And these are things that are tough to deal with in COVID. And we have more so-called objective studies of PCR tests. So they're taking multiple PCR tests. What happens if you take the PCR test and you've had special water? What happens if you take the PCR that if you haven't, you just take ordinary water? And we compare the two. Very significant. No significant differences. I'm looking for my pointer again. Headache, fatigue, aches, eyes, shortness of breath were not statistically significant different whether you had the treated water or the untreated water. And I think I know why. The headache, the fatigue, the aches in the eyes, getting regular medical care only produced a small increase in symptoms with the COVID, and they started to resolve on ordinary medical care. Since they were already quickly resolving on ordinary medical care, to throw in the drops doesn't add anything. In the areas in COVID where there's a serious effect, overall, how do you feel? Overall temperature, the lingering sore throats, the coughs, and of course the objective PCR positivity. This effect starts very, very quickly. Actually, this is time sequence a little bit differently, but after about a day, although here on this chart it says day 2, but different meaning. After about a day, the groups split off. And the ones taking the special drops are getting better faster. The ones not taking the special drops, they're still getting better. So we're not going to COVID to die, but you're going there to try to resolve within a certain length of time in the hospital. Does the hospital eventually resolve? Yes. Does it resolve faster if you've had the drops? Without question. And you can see this with error bars and all of that kind of good stuff. We don't need to go geek on this. So if we go back to the central question, along the x-axis, we have scalability. Along the y-axis, we have effectiveness. So high effectiveness, low scalability would be something like you saw in one person treating one person. Let's assume they fix that person. So what? There's an inexhaustible supply of people in pain. So hands-on healing, or its analog, think of the cousins. The hands-on healing presumably has high effect size. I mean, I certainly have found it, but it doesn't make it scalable. Cotton and treated water is a little more scalable and is also very highly effective. And we come over here to high scalability, but we have, using this is only qualitative, resonant water, which is the stuff I just got finished talking about, that seems to be scalable, and it seems to be maybe even more effective than the original. The recording has high scalability, but it doesn't seem to be as strong. We've tried other methods of recording, which I'm not dealing with here, including ordinary audio sound studios, to see what happens. And that also produces significant genomic changes, but not as much as the fancy one in the Faraday cage, which doesn't produce as strong an effect size as live. So if you're looking at this, the most promising by far is the resonant water. Because it's not maximally, but it's close to maximally scalable, but it's also really high in terms of effects. So we need to fill in this chart and find out what works, what doesn't, what, in terms of scalability and storability. And we can look at this, think of all the possibilities clinically, think of all the possibilities experimentally. What would happen if you were a healer, you supplied clients with treated water? How did they interact? How did method A interact with method B? Very little is known about this. I'm starting some more formal experiments. So you get the idea. I'm going to try to bring this on home. I got to thank a bunch of people who've put up with me, including Brown University, IONS, Tokyo University, Beech Tree Labs, Emerald Gate. I leave you with a cartoon just that I think is funny. You may not find it funny. And I say, thank you. I'm going to end my sharing and see if there are any questions.